everybody. We completed photosynthesis in the previous class. Now we take respiration, nitrogen metabolism up to biological nitrogen fixation. We all know photosynthesis is an anabolic process where two complete inorganic components combine to form an organic component to liberate oxygen. Respiration is a catabolic process where oxygen is used, carbohydrates are broken into carbon dioxide and water, and large amount of energy is liberated. That energy liberated is stored as energy currency of the cell called ATP. ATP structure is, ATP is almost a nucleotide. It shows three parts: nitrogen base, sugar, phosphate. Nitrogen base in ATP is adenine. Sugar is ribose sugar. So, definitely it is RNA. Phosphates are alpha, beta, gamma. So, when you write this, adenine, ribose, alpha, beta, gamma. In between beta and gamma, the bond is very strong bond. When you break this bond, the amount of energy liberated is 7.6 kilocalories. So, 1 ATP is equal to 7.6 kilocalories. By hydrolyzing ATP, we get 7.6 kilocalories. Like this, ATP is stored and supplied to various cell organelles whenever it is required. And aerobic respiration, anaerobic respiration. Respiration in the presence of oxygen is aerobic. In the absence of oxygen is anaerobic. Aerobic occurs in 4 steps: glycolysis, oxidative decarboxylation of pyruvic acid, Krebs cycle, electron transport system. In total, 6 oxidations occur. 1 in glycolysis, 1 in between glycolysis and Krebs cycle, that is oxidative decarboxylation of pyruvic acid in mitochondrial matrix, and 4 in mitochondrial matrix during Krebs cycle. All these reduced coenzymes NADH2, FADH2 involve in electron transport system to produce ATP, which is also called oxidative phosphorylation. Then anaerobic respiration, where fermentation occurs and alcohol is formed, and RQ, respiratory quotient. All this we discuss in respiration. We discuss some bits in respiration first. Based on RQ values, arrange the following in ascending order: oxalic acid, glucose, tripalmitin, tartaric acid, malic acid, proteins. Oxalic acid, tripalmitin are fats, so less than 0, less than 1. Proteins are also, oxygen content is less, carbon dioxide is more, so during respiration more oxygen is absorbed and less carbon dioxide is liberated. So, answer will be tripalmitin first, then proteins, then glucose, then malic acid 1.33, then tartaric acid 1.4 or something like that, and 1 oxalic acid is 6. Oxalic acid is 6. So, last is oxalic acid. Answer is 3, 6, 2, 5, 4, 1. So, tripalmitin, protein, glucose, malic acid, tartaric acid, oxalic acid. So, based on RQ values, arrange the following in ascending order is first answer. The ratio between the number of ATP generated by substrate level phosphorylation in hexose phosphate pathway. Hexose phosphate pathway is nothing but glycolysis. Hexose phosphate pathway, and number of water molecules formed during ETS per 1 glucose molecule. Substrate level phosphorylations are 2. Number of water molecules liberated during electron transport per glucose molecule is 6. So, 2 is to 3, 1 is 2 is to 6, 1 is to 3. So, 2 is to 6, 1 is to 3. So, the ratio between the number of ATP generated by substrate level phosphorylation in hexose phosphate pathway. Hexose phosphate pathway is nothing but glycolysis. Hexose phosphate pathway, and number of water molecules formed during electron transport system per 1 glucose molecule is 2 is to 6, which is nothing but 1 is to 3. Next, electron transport complex with copper containing proteins. Electron transport complex with copper containing proteins. Complex 1 NADH dehydrogenase complex contain many iron sulfur proteins. Complex 2 ferredoxin, that means succinic dehydrogenase complex, contain 2 iron sulfur proteins. Complex 3 cytochrome C oxidase will contain 1 iron sulfur protein. Complex 4, which is called as cytochrome C oxidase, always contain 2 copper containing proteins. So F0 F1 ATPase is coupling factor complex. Ubiquinone is a mobile carrier between complex 1 and complex 3, complex 2 and complex 3 during electron transport system. Cytochrome reductase is nothing but complex 3 with 1 iron sulfur protein. So electron transport complex with copper containing proteins is cytochrome C oxidase. It is also called as complex 4, cytochrome C oxidase or complex 4, third option. Substrate which undergoes reduction in anaerobic respiration in yeast. Substrate which undergoes reduction when pyruvic acid undergoes decarboxylation during this one, acetaldehyde is formed. CH3 CHO. This acetaldehyde utilizes the NADH2 formed in glycolysis and converts into ethyl alcohol. So acetaldehyde is undergoing reduction to produce ethyl alcohol during fermentation. So pyruvic acid by decarboxylation produces acetaldehyde, because pyruvic acid is CH3CO COOH. The CO will go and what it becomes CH3 CHO, and ultimately it becomes C2H5OH. So pyruvic acid by decarboxylation produces acetaldehyde. Acetaldehyde undergo reduction and produces what you call ethyl alcohol. So substrate which undergoes reduction in anaerobic respiration of yeast cells is alpha ketoglutarate is wrong, ethyl alcohol is wrong, formaldehyde is wrong, acetaldehyde is correct. So fourth question, second option. During glycolysis, the ratio of ATP consumed, NADH2 formed and ATP liberated. The ratio of ATP consumed is 2, NADH2 formed is 2, ATP formed is 4. So 2:2:4, which is nothing but 1:1:1. 2:2:4 is 1:1:2. So during glycolysis, the ratio of ATP consumed 2, NADH2 formed 2, ATP liberated 4, 2:2:4, that is 1:1:2. So third option will be for this. Now study the following table, select the correct pair of answers in which the former is the pair shows the set of reactions that do not take place during glycolysis, and the latter is the pair shows the set of reactions taking place during Krebs cycle. Decarboxylation, condensation, hydration. Isomerization, dephosphorylation, hydration. Dehydration, condensation, decarboxylation. Condensation, decarboxylation, isomerization. So select the correct pair of answers in which the former in the pair shows the set of reactions that do not take place during glycolysis. Decarboxylation does not occur, condensation does not occur, hydration does not occur. Set of reactions that do not take place during glycolysis, and the latter in the pair shows that set of reactions that takes place during Krebs cycle. Dehydration, condensation, decarboxylation occurs during Krebs cycle. Decarboxylation, condensation, hydration does not occur during glycolysis. So answer is 1 and 3. 1 and 3, decarboxylation, condensation, hydration does not occur during glycolysis. Dehydration, condensation, decarboxylations occur during Krebs cycle. In respiratory electron transport, which of the following acts as both as electron and proton carrier? As electron and proton carrier, complex 3 is wrong, cytochrome C is wrong, complex 2 is wrong, ubiquinone is correct, because ubiquinone is a mobile carrier between complex 1 and complex 3, complex 2 and complex 3. This ubiquinone takes electrons from either FAD, FMN, flavin mononucleotide prosthetic group of complex 1, or FAD, flavin adenine dinucleotide, that is prosthetic group of complex 2. It takes electrons. Whenever it takes electrons, it takes 2 protons from mitochondrial matrix. So 2 protons plus 2 protons, 4 protons are transferred from mitochondrial matrix to intermembrane space or intermitochondrial space. So in respiratory electron transport system, which of the following acts as both electron and proton carrier? Is definitely ubiquinone, because it takes electrons from complex 1 or complex 2. Immediately by taking protons, it converts into ubiquinol. Ubiquinol liberate electrons into the complex 3, and again become ubiquinone, which resembles the quinone cycle of photosynthesis. So, in respiratory electron transport, which of the following acts as both electron and proton carrier is ubiquinone? Third option. Isocitric acid is the substrate for first oxidation of Krebs cycle, first decarboxylation step in mitochondrial matrix, third oxidation reaction in aerobic respiration, second oxidation reaction in mitochondrial matrix. First oxidation is Krebs cycle is correct. Isocitric acid converts into oxalosuccinic acid. First decarboxylation step in mitochondrial matrix is wrong because oxalosuccinic acid is answer for this. Third oxidation reaction in aerobic respiration. Total third oxidation because one in glycolysis, one in between glycolysis and Krebs cycle. So, when it comes to the total one, this is third oxidation reaction in aerobic respiration and second oxidation reaction in mitochondrial matrix. Definitely, first oxidation reaction in mitochondrial matrix is oxidative decarboxylation of pyruvic acid. Second is this one. So, answer is 1, 3 and 4 are correct. 1, 3 and 4 are correct. This is an assertion reasoning question. Oxalic acid has more RQ value than malic acid. Oxalic acid RQ value is 4, malic acid RQ value is 1.33. Oxalic acid needs more number of oxygen molecules than malic acid for its oxidation. So, oxalic acid has RQ value more than malic acid is correct. Oxalic acid needs more number of oxygen molecules than malic acid for its oxidation is wrong. So, A is true or is false. A is true or is false. The number of ATP formed through substrate level phosphorylation and FADH2 oxidation in 2 turns of Krebs cycle. 2 turns of Krebs cycle means number of ATP per each Krebs cycle is 1. This forms when succinyl coenzyme A converts into succinic acid. Then when succinic acid is converts into fumaric acid, one NADH2, one FADH2 is formed. So, here one ATP is formed, here one F. When FADH2 participates in electron transport system, it is equivalent to 2. So, 2 plus 2 for every 2 turns it is asked. Number of ATP formed through substrate level phosphorylation and FADH2 oxidations in 2 turns of Krebs cycle will be, for 1 turn it is 3. For 2 turns it is 6. So, the number of ATP formed in substrate level phosphorylation and oxidation of FADH2 or whatever it may be, number of ATP formed through substrate level phosphorylation and FADH2 oxidations in 2 turns of tricarboxylic acid cycle. Tricarboxylic acid cycle is nothing but Krebs cycle. Answer is not 3, 4, 5, it is 6. The only 5 carbon intermediate compound of TCA cycle is the precursor of the following amino acid. The only 5 carbon intermediate compound of TCA cycle is alpha ketoglutarate acid. It is the substrate which is used as a precursor in the formation of following amino acid is glutamic acid. So, glutamic acid is formed from alpha ketoglutarate when aspartic acid combines with alpha ketoglutarate acid, or when alpha ketoglutarate acid converts. The only 5 carbon intermediate of tricarboxylic cycle is the precursor of the following amino acid, means from which amino acid metabolism takes place, and glutamic acid formed is alpha ketoglutarate acid. So, glycine, serine, glutamic acid, plastosemiquinone. Answer is glutamic acid. Identify the correct statement about succinic dehydrogenase. It involves in the oxidation of substrate but not in the decarboxylation. Correct. It is the enzyme of third oxidation step in TCA cycle. Correct, because when succinic acid is converted into fumaric acid, this is involved. It is the enzyme of fifth biological oxidation in the total aerobic respiration. Correct. It is the enzyme of fourth oxidation step in mitochondrial matrix. Correct. And NAD plus is the coenzyme for this whole enzyme is wrong. It is FAD plus. So, answer is A, B, C, D. So, identify the correct statements about succinic dehydrogenase enzyme regarding Krebs cycle is it involves in Oxidation of substrate but not in decarboxylation, correct. It is the enzyme of third oxidation step in tricarboxylic acid cycle is correct from pyruvic acid. It is the enzyme of fifth biological oxidation starting from glucose in cytosol. It is the enzyme for fifth biological oxidation in aerobic respiration. It is the enzyme of fourth oxidation step in mitochondrial matrix. Definitely starting from pyruvic acid, it will be fourth oxidation step. Now, NAD plus is the coenzyme for this holoenzyme is wrong. NAD plus is not the coenzyme of this holoenzyme. FAD plus is the coenzyme of this holoenzyme. So, A, B, C, D are correct. E is wrong. Fourth option. This is an assertion reasoning. Oxidation of each molecule of cytosolic NADH results in the synthesis of only 2 ATP in mitochondria. Because the complex 1 does not involve extra 4 protons will not come, so only 6 protons will come. 6 protons when moves through a coupling factor complex and show chemiosmotic theory, they are equivalent to 2 ATP. So, oxidation of each molecule of cytosolic NADH2 results in the synthesis of only 2 ATP in mitochondria, correct. Each cytosolic NADH2 on oxidation transfers its electrons directly to ubiquinone pool, so that 6 protons 4 plus 2 only transferred to the perimitochondrial space or intermitochondrial space. Because only 6 protons are transferred to the intermitochondrial space or perimitochondrial space, it is equivalent to 2 ATP. So, A and R are correct. R is the correct explanation of A. A and R are correct. R is the correct explanation. Oxidation of each molecule of cytosolic NADH2 results in the synthesis of only 2 ATP in mitochondria is correct. Each cytosolic NADH2 on oxidation transfers its electrons directly to ubiquinone pool, so that 6 protons 4 plus 2 only are transferred to the perimitochondrial space is correct. So, A and R are correct. R is the correct explanation of A. Impermeability of inner mitochondrial membrane to protons is advantageous to the cell. Because impermeability is there, protons move through coupling factor complex, and when they reach the smallest rotator of the universe CF1, for every 3 protons 1 ATP is formed. So, impermeability of inner mitochondrial membrane to protons is advantageous to the cell is correct. This property helps in establishing a transmembrane proton gradient, which ultimately helps in ATP synthesis. Impermeability to inner mitochondrial membrane to protons is advantageous to cell is correct. Why? This property helps in establishing a transmembrane proton gradient, which ultimately helps in ATP synthesis, correct. So, both A and R are correct. R is the correct explanation of A, first option. Cytochrome C is a mobile carrier between these complexes. Cytochrome C, so this is complex 3 and this is complex 4. In complex 3, cytochrome B with 560, 565 is present. This is complex 3, cytochromes reductase. This is complex 4, cytochrome C oxidase. And cytochrome C1 with 1 iron sulfur protein. Complex 4, cytochrome A1, cytochrome A3, 2 copper containing proteins. Whenever electrons come here and want to go, they are taken by a special mobile carrier called cytochrome C. This mobile carrier transfers electrons like this, and finally they are added to half O2. So, the mobile carrier that is present in between complex 3 and complex 4. Cytochrome C is a mobile carrier between these complexes, means definitely 3 and 4. Ubiquinone is a mobile carrier between complex 1 and complex 3, complex 2 and complex 3. Cytochrome C is not a mobile carrier between 2 and 3, 1 and 3, 2 and 4, but it is in between cytochrome C oxidase, cytochrome C reductase complex and cytochrome C oxidase complex, which are nothing but complex 3 and complex 4. So, answer is 3. In Krebs cycle, FAD involved in the formation of, whenever succinic acid, a 4 carbon compound, undergoes oxidation in the presence of succinic dehydrogenase, one FADH2 is liberated and fumaric acid is formed. So, from succinic acid to fumaric acid, in the presence of an enzyme succinic dehydrogenase, FADH2 is formed. Succinic dehydrogenase is a very important bit. It is the only enzyme which is not present in mitochondrial matrix but present in the intermitochondrial space. So, in Krebs cycle, FAD is involved in the formation of fumaric acid, second one. In glycolysis, hexose is cleaved into two trioses by this enzyme. Kinase is wrong, dehydrogenase is wrong, enolase is useful for dehydration step, aldolase. Aldolase converts fructose 1,6 biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So, fructose 1,6 biphosphate 6 carbon compound converts into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate in the presence of an enzyme aldolase. These two are called triose phosphates. These two are also interconvertible in the presence of glyceraldehyde 3-phosphate isomerization. Glyceraldehyde 3-phosphate isomerase. So, aldolase is the enzyme which converts a 6 carbon compound fructose 1,6 biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are nothing but triose phosphates. Correct the sequence of the 4 steps of aerobic respiration. Oxidation of NADH2 FADH2, formation of CO2 NADH2 FADH2, formation of acetyl coenzyme A, glucose converting into pyruvic acid. So, always first glucose converting into pyruvic acid is glycolysis. Conversion of pyruvic acid and formation of acetyl CoA is definitely oxidative decarboxylation of pyruvic acid in mitochondrial matrix, which is nothing but link reaction. Then formation of CO2 NADH2 FADH2 is Krebs cycle, and oxidation of NADH2 and FADH2 is electron transport system. So, glycolysis, link reaction, Krebs cycle, electron transport system, DCBA. This is the correct sequence during aerobic respiration. This is an assertion reasoning. Acetyl coenzyme A is popularly known as connecting link between glycolysis and Krebs cycle. Definitely, acetyl coenzyme A is regarded as a connecting link between glycolysis and Krebs cycle. Pyruvic acid formed as the end product of glycolysis enters the Krebs cycle only through acetyl coenzyme A. So, pyruvic acid which is formed in glycolysis, 3 carbon compound, undergoes oxidative decarboxylation. First it undergoes decarboxylation, then into oxidation in the presence of pyruvate dehydrogenase complex, converts into acetyl coenzyme A, 2 carbon. This is the connecting link between glycolysis and Krebs cycle. 6 cofactors helps in this process: NAD plus, FAD plus, coenzyme A, magnesium, thymine pyrophosphate and lipoic acid which is oxidized. Pyruvate dehydrogenase is not a single enzyme, it is a combination of 3 enzymes: dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase and pyruvic decarboxylase. This is called link reaction, and in the link reaction pyruvic acid converts into acetyl CoA. Only after the formation of acetyl CoA aerobic respiration perfectly occurs, Krebs cycle occurs. So, acetyl coenzyme A is popularly known as connecting link between glycolysis and Krebs cycle. Pyruvic acid formed at the end of as end product of glycolysis enters the Krebs cycle only through acetyl coenzyme A. Both A and R are correct. R is the correct explanation of A. Assertion: RQ value of fats is less than 1. Definitely RQ value of fats is less than 1. Why? Because fats contain less oxygen and hence consume more oxygen during their oxidative breakdown. In the chemical structure of fats, oxygen amount will be less, carbon dioxide amount will be less. Whenever oxygen amount is less during respiration, more oxygen is absorbed and less carbon dioxide is released. What is RQ? CO2 released by oxygen absorbed. So whenever CO2 is less and oxygen is more, definitely its value is less than 1. So both A and R are correct. R is the correct explanation of A, first option. This is another assertion reasoning. Glycolysis may be considered as an aerobic part of anaerobic respiration. Glycolysis is a common pathway for aerobic and anaerobic respiration methods. Glycolysis cannot be considered as an anaerobic because anywhere in glycolysis oxygen is not used. So glycolysis may be considered as an aerobic part of anaerobic respiration is wrong. But glycolysis is the common process or common step for both aerobic and anaerobic respirations is correct. So A is false, R is true, fourth option. Study the following list. List 1 biochemical reaction, list 2 enzyme. Conversion of 3-phosphoglyceric acid to 2-phosphoglyceric acid. Cis-aconitic acid to isocitric acid. 1,3-biphosphoglyceric acid to glyceraldehyde 3-phosphate. Xylose 5-phosphate to ribulose 5-phosphate. Conversion of glyceraldehyde 3-phosphoglyceric acid to 2-PGI is by mutase, phosphoglyceromutase. This is popularly called intramolecular shift. Fourth option. Cis-aconitic acid to isocitric acid is by aconitase. Aconitase helps in hydration and dehydration reactions. 1,3-biphosphoglyceric acid to glyceraldehyde 3-phosphate is dehydrogenase. It is dehydrogenase and kinase. Then xylose 5-phosphate to ribulose 5-phosphate. It is by epimerization. So answer is A4, B3, C5, D1. Study the following table. Substrate, enzyme, product. Glyceraldehyde 3-phosphate, isomerase, dihydroxyacetone phosphate. Here we have to identify incorrect common. Ribulose 5-phosphate, epimerase, ribulose 5-phosphate. Glucose, hexokinase, glucose 6-phosphate. Fumaric acid, enolase, malic acid. Glyceraldehyde 3-phosphate in the presence of isomerase converts into dihydroxyacetone phosphate is correct. Ribose 5-phosphate in the presence of epimerase, ribulose 5-phosphate is wrong. It is not epimerase, it is isomerase. Glucose in the presence of hexokinase converts into glucose 6-phosphate is correct. Fumaric acid in the presence of enolase converts into malic acid is wrong. Fumaric acid in the presence of fumarase undergo hydration and produces malic acid. So wrong combinations are 2 and 4. Assertion. Oxidation of succinic acid to fumaric acid occurs in cytosol. Oxidation of succinic acid to fumaric acid occurs in cytosol is wrong. It occurs in mitochondrial matrix. Why? This is the process which is occurring in mitochondrial matrix, which is nothing but TCA cycle or Krebs cycle. Succinic dehydrogenase reduces FAD. Succinic dehydrogenase reduces FAD is correct, but oxidation of succinic acid to fumaric acid occurs in cytosol is wrong. So A is false, R is true. Fourth option. How many ATP are produced as net gain through oxidative phosphorylation from one glucose molecule if the last two oxidations in TCA cycle fails to occur? If the last two oxidations means one NADPH2, one NADH2 and one FADH2. When NADH2 and FADH2 are not formed, so what is the ATP formula? How many ATP are produced as net gain through oxidative phosphorylation from one glucose molecule if the last two oxidation in TCA cycle fail to occur is 22 ATP. 22 ATP are formed. First option. Total number of ATP formed as a result of complete breakdown of one molecule of pyruvic acid. Complete breakdown of one molecule of pyruvic acid. How many ATP molecules are formed is total Krebs cycle takes place, electron transport takes place, it is equivalent to 16 ATP. RQ value varies with the nature of respiratory substrate. RQ value differs with the nature of respiratory substrate is correct. Definitely, according to the oxygen percentage, carbon dioxide percentage in the chemical structure, RQ value is decided. Reason. the proportion of carbon oxygen is not the same in different respiratory substrate. Definitely the proportion of carbon oxygen is not that same in different respiratory substrate. That's why the RQ value varies in nature of various respiratory substrates. So both A and R are correct. R is the correct explanation of A. Another assertion reasoning. Respiration is an exergonic process. Respiration is an exergonic process is correct. That means energy is liberated out. ATP formed during respiration on hydrolysis yield energy. So respiration is an exergonic process is correct. ATP is formed during respiration. If it is stopped there, the answer will be first. But here ATP formed during respiration on hydrolysis yield energy means whenever ATP formed anywhere, it yields energy by hydrolysis, not because respiration is an exergonic process. So here both A and R are correct. R is not the correct explanation of A. Second option. Many assertion reasons are given this time because of their importance. Synthesis of ATP in glycolysis is called as substrate level phosphorylation. Definitely, when 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid, 1 ATP is produced, that is substrate level phosphorylation. When pyruvic acid is converted into, or phosphoenol pyruvic acid is converted to pyruvic acid, 1 ATP is formed, that is another substrate level phosphorylation. Energy rich phosphorus necessary for ATP formation is taken from the substrate of glycolysis. So because the phosphates are taken from the substrate of glycolysis, it is called substrate level phosphorylation. So A and R are correct. R is the correct explanation of A. So assertion A, synthesis of ATP in glycolysis is called substrate level phosphorylation, correct. Energy rich phosphorus necessary for ATP formation is taken from the substrates of glycolysis, correct. So both A and R are correct. R is the correct explanation of A. Number of hydrogen translocated in the intermembrane space through different complexes according to their sequence for the oxidation of each mitochondrial NADH2. Whenever mitochondrial NADH2 involves, first it passes through complex 1. Whenever it passes through complex 1, 4 protons are located here. Then ubiquinone takes 4 protons from here. Ubiquinone takes 4 protons from here and makes stored here. Then different cycles takes place. Ultimately it is added to half O2. 2 protons are added to half O2 resulting in the formation of water. 2 protons move through the complex 4 to become 4 here. So whenever NADH2 of mitochondrial matrix involved in the process, the protons stored there are 4 plus 4 plus 2. That means 10 protons. So whenever complex 1 involves, 4 protons in addition moves to intermitochondrial space. So whenever the total cycle is over by ubiquinone cycle 4, from complex 4 to, so in total number of protons translocated into the intermembrane space through different complexes according to their sequence for the oxidation of each mitochondrial NADH2 is 4 plus 4 plus 2, 10 protons. The correct split up of 6 oxidations which occur in different steps of aerobic respiration, 1 glucose molecule. Correct split up is 1 in glycolysis, 1 in between glycolysis and Krebs cycle, 4 in Krebs cycle. So correct split is 1, 1, 4. How? 1 in glycolysis, 1 in between glycolysis and Krebs cycle, which is also occurs in mitochondrial matrix, but conversion of pyruvic acid to acetyl coenzyme A. 4 in Krebs cycle. So answer is fourth one. 1, 1, 4. More CO2 is evolved than the consumed oxygen in the, if the respiratory substrate is. More CO2 is evolved than the consumed oxygen if the respiratory substrate is. If it is glucose, only 1. If it is tripalmitin, less than 1. If it is oleic acid, less than 1. Then when you get more than 1, whenever you get an RQ value more than 1, then this statement will come into existence. What is that? More carbon dioxide is evolved than the consumed oxygen if the respiratory substrate is tartaric acid. If the respiratory substrate is tartaric acid. Role of oxygen in the process of aerobic respiration. Role of oxygen in the process of aerobic respiration. Role of oxygen is in aerobic respiration. 1. To donate electrons, wrong. 2. To accept electrons. So, during electron transport system, it accepts electrons. Then, to donate protons, wrong. To accept protons is correct. By taking 2 electrons from complex 4, by taking 2 protons from the process, water is produced. So, the role of oxygen in the process of aerobic respiration is to donate electron is wrong. To accept electrons and to accept protons is correct. So, answer will be B and D. Answer is B and D. Which of the following serves as respiratory substrate only during starvation? Which of the following serves as respiratory substrate only during starvation means when the food is not available? Malic acid is wrong, triolin is wrong, starch is wrong, but proteins. They definitely serves as a respiratory substrate only during starvation. Proteins, third option. In glycolysis, water molecule is removed from. In glycolysis, water molecule is removed from means whenever 3-PGA converts into 2-PGA, it is intramolecular shift occurs in the presence of phosphofructoisomerase or phosphoglyceromutase. Then 2-PGA converts into phosphoenol pyruvic acid in the presence of an enzyme enolase. In the presence of an enzyme enolase. So, this is a dehydration step where water is removed. So, 3-PGA converts into 2-PGA intramolecular shift. 2-PGA converts into phosphoenol pyruvic acid. This is dehydration which occurs in the presence of enolase. So, in glycolysis, water molecule is removed from which one? 2-phosphoglyceric acid to form phosphoenol pyruvic acid. In Krebs cycle, the substrate for final oxidation step is the product of the following biochemical reaction. What is the final one? Malic acid. How this malic acid is formed? By the dehydration or hydration of fumaric acid. So, fumaric acid by removing water molecule or by adding water molecule converts into malic acid. So, in the Krebs cycle, the substrate for the final oxidation step is the product of the following biochemical reaction: hydration. Whenever water is added to fumaric acid, it converts into malic acid. Malic acid undergoes Locke's oxidation and produces oxaloacetic acid, which combines with acetyl coenzyme A, and the process continues. So, in Krebs cycle, the substrate for final oxidation step is the product of the biochemical reaction means hydration, not condensation or decarboxylation, because by condensation citric acid is formed. By decarboxylation, alpha ketoglutarate acid is formed. By dehydration, cis-aconitic acid is formed. So, answer is hydration, fourth one. Number of ATP molecules formed through substrate level phosphorylation in glycolysis and Krebs cycle during the complete oxidation of one glucose molecule. In glycolysis, whenever 2 molecules of 1,3-diphosphoglyceric acid becomes 2 molecules of phosphoglyceric acid, 2 ATP. When 2 molecules of phosphoenol pyruvic acid converts into pyruvic acid in the presence of pyruvate kinase, 2 ATP. So, in total by substrate level phosphorylation by glycolysis, 4 ATP are formed. In Krebs cycle, when succinyl coenzyme A, 2 molecules of succinyl coenzyme A, 1 molecule for 1 pyruvic acid, 2 molecules for total glucose. Whenever 2 molecules of succinyl CoA converts into succinic acid, 2 ATP are formed. So, number of ATP molecules formed through substrate level phosphorylation in glycolysis and Krebs cycle during the complete oxidation of 1 glucose molecule, 4 in glycolysis, 2 in Krebs cycle, third option. This is another assertion reasoning. Clostridium botulinum is an obligate anaerobe. So, anaerobes are divided into 2 types: facultative anaerobes, obligate anaerobes. Obligate anaerobe means which cannot survive in the presence of oxygen. Facultative anaerobe means it can survive in the presence of oxygen. Clostridium is an obligate anaerobe. Yeast cell, Saccharomyces cerevisiae are facultative anaerobe. So, Clostridium botulinum is an obligate anaerobe is correct. It cannot survive in the absence of oxygen is wrong. It cannot survive in the presence of oxygen. So, A is true or is false, third option. The incorrect about glycolysis. Oxygen utilized in the beginning, CO2 liberated at the end, 2 ATP net gain, no decarboxylation. 2 ATP net gain is correct. No decarboxylation. Anywhere carbon dioxide is not liberated. So the incorrect about glycolysis or oxygen utilized in the beginning is wrong. CO2 liberated at the end is wrong. So in glycolysis, important steps are common for both aerobic and anaerobic. Common for both aerobic and anaerobic. CO2 is not used. O2 is not liberated. Occurs in cytosol. Glucose always produces 2 pyruvic acids and always 2 NADH2 and 2 ATP are formed. So these are the important steps that occurs during glycolysis. So here the answer is carbon dioxide is liberated at the end is wrong. Oxygen utilized in the beginning is wrong. Second option. Number of ATP formed due to oxidation of 2 cytosolic NADH2, 8 mitochondrial NADH2 and 2 FADH. Cytosolic NADH2, each NADH2 is equal to 6 protons, so 2 ATP. So cytosolic NADH2, 2 into 2, 4. 8 mitochondrial NADH2, each NADH2 10 protons, so 3 ATP, 8 into 3, 24. And FADH2 respectively, FADH2 also 6 protons, 2 ATP, so 2 FADH2, 4. So 4, 24 and 4. 4, 24 and 4, second option. How many ATP are obtained in the reaction which involves in the formation of one molecule which is often described as connecting link between glycolysis and Krebs cycle? Whenever connecting link between glycolysis and Krebs cycle occurs, pyruvic acid converts into acetyl coenzyme A. Whenever pyruvic acid acetyl coenzyme A, pyruvic acid converts into acetyl coenzyme A, they are equivalent to 1 NADH2. Whenever 1 NADH2 involves, the NADH2 is of mitochondrial matrix. So 10 protons means 3 ATP. So how many ATP are obtained in the reaction which involves in the formation of one molecule which is often described as connecting link between glycolysis and Krebs cycle is 3 ATP from 1 NADH2. Study the following and identify the correct combination. Substrate glucose, nature carbohydrate, RQ value. Glucose, carbohydrate 1 is correct. Oleic acid, fat 1.33 is wrong. More than 1 means it is not a protein or fat, it must be organic compound. So oleic acid, fat 1.33 is wrong. Malic acid, organic acid 0.7 is wrong. Whenever it is organic acid, its alkyl value is more than 1 because the oxygen content is more and carbon dioxide content is less. Definitely oxygen used is less and carbon dioxide produced is more. So definitely malic acid, organic acid is correct, but 0.7 is wrong. Tartaric acid, organic acid 1.6 is correct. So answer is 1 and 4. Glucose, carbohydrate, RQ value is 1. Oleic acid, fat 1.33 is wrong. It is a organic acid, it is a fat. Malic acid, organic acid 0.7 is wrong. Tartaric acid, organic acid 1.6 is correct. So 1 and 4 is correct answer. Ubiquinone is a mobile electron and proton carrier, correct. Ubiquinone is a lipid soluble quinone. Because it is a lipid soluble quinone, it is not taking electrons and protons. It is nature is taking electrons and protons from complex 1 to 3 and complex 2 to 3. So ubiquinone is a mobile electron and proton carrier is correct. Ubiquinone is a lipid soluble quinone is correct, but both are not linked. So A and R are correct. R is not the correct explanation of A. Second option. Two membranes which has special role in energy transduction. Two membranes which has special role in energy transduction means formation of ATP. Membranes of Golgi complex wrong. Thylakoid membrane is correct. Thylakoid membrane only allows the movement of protons, does not allow the movement of protons. So involves coupling factor complex which helps in ATP forming. Inner mitochondria, inner membrane of mitochondria is also impermeable to protons, that's why helps in the formation of ATP. Tonoplast which is present around vacuole is also wrong. So answer is 2 and 3. The membranes which has special role in energy transduction are thylakoid membrane in chloroplast, inner membrane of mitochondria in mitochondria. One involves in photosynthesis, results in the formation of ATP. Other involves in respiration, results in the formation of ATP. So 2 and 3 is the answer. Next, assertion reason. Assertion. Respiration is concerned only with the release of energy. Respiration is concerned only with the release of energy is correct, because oxidation, whenever occurs, energy is liberated. Reason. During respiration, the sugar molecules are also modified to form the carbon skeletons that make up the basic building blocks of cell structure. In respiration, the sugar molecules are not modified, they are broken, oxidized to produce some intermediate compounds that form carbon skeletons that make up basic building blocks of cell structure. So A is correct, R is false. A is false.